Hi everyone, it's Devin Jensen from BD Biosciences. I'm back to show you the exciting new developments in version 2 of our BD Cellismo data visualization tool. We've added support for the BD Rhapsody single-cell attack-seq assay and much more. Let's jump in and take a look.
First, batch correction. The goal of batch correction is to remove unwanted technical variation between batches while hopefully preserving true biological differences. Now, the Rhapsody system can have very low batch effects, as you can see from this example, 3 replicates of the same PBMC sample. So in order to show you batch effect correction in Cellismo, we're going to look at a more extreme example of 2 different versions of our whole transcriptome assay. These 2 datasets from the same donor are showing large batch effects due to the difference in sensitivity of the improved assay, even though they have the same cell types. To fix that, we can go to Utility Tools, Batch Correction page, select the variable on which we want to correct, and press the button. Now what's happening is that we're estimating and removing the technical effects of that batch correction based on that variable. And when that's complete, we'll have a new layer of information with batch corrected counts. Once it's finished, you're going to want to create a new set of dimensionality reduction coordinates based on the batch corrected counts. We've already done that, and you can see the results here. Much better. Once you have batch corrected counts, you can toggle between the original counts and the batch corrected counts with this button here. For integration, the goal is to combine multiple datasets into a common coordinate system that allows you to do clustering or visualization. Here's 2 samples from 2 different donors. If we go to the Utility Tools Integrate Data tool, we can select a variable for integration and click the button. Once that's done, again, you're going to want to create new dimensionality reduction coordinates. We've already done that, and I can show you the result here. Within the quality control section, there's a new doublet detection tool. Let's click to run it. Now, because this dataset has absec markers, I happen to be able to see that there are some doublets. Let's look at CD14 versus CD3 absec. And there's a group of double positive cells up here. Generally, that doesn't happen. If we look on the dimensionality reduction scatter plot, we can see that they're focused in this cluster here. Let's see what the doublet detection tool came up with based on RNA. All right, let's view the results. Yep, that cluster is a doublet cluster. We can easily remove it with this button here. Now, in adding support for the attack-seq assay, we've made some changes to the BD Rhapsody sequence analysis pipeline as well. For the best experience, you're going to want to run attack-seq data in version 3.0, which will create new modalities in the Cellismo output file. The attack peaks modality, attack gene activity, which is often used as a proxy for RNA, so this is less important if you already have RNA in a multi-omic experiment, and also the transcription factor attack motif by cell matrix. Here we have a mouse kidney nuclei dataset. It's multi-omic with whole transcriptome and attack-seq data. You can see the RNA modality and the 3 attack-seq modalities. The first thing we'll do is do a cell classification with a new algorithm we've included called sctype. This allows you to provide a custom CSV file of mouse kidney markers, positive or negative. It also requires a categorical cell annotation, so you'll want to do some clustering first. Let's see those results. Next, we're going to take a closer look at this loop of Henle thick ascending limb cell type. Let's do some differential analysis on those cells. So we'll select that cell type versus all the rest, and we want to look at the attacks peaks modality. Let's start that analysis. Okay, we've got our result. What I'm going to do is take this box selection mode and choose the peaks that are significantly up in our thick ascending limb cells. So we'll just take this chunk of peaks, create a set out of those based on those currently selected in the graph, and call it TAL peaks up. Now with this set of peaks, I can come over here to the attack tools and choose motif enrichment analysis. I'll choose to do this with the set of peaks that we just created, and start that analysis. Now what it's going to do is look at the sequence of the DNA within those peaks and look for motif signatures that are enriched in those peaks compared to a set of background peaks. And we'll get back to the list shortly. The table of results show the transcription factor motifs enriched in our set of peaks. One of the top results by p-value is this ESRRB motif. Let's look at that on the dimensionality reduction scatter plot. It is indeed enriched in the cell's loop of Henry thick ascending limb. This result perfectly matches a separate publication from a separate mouse kidney dataset, which notes that the loop of Henry cells are enriched in the motif for ESRRB. Another tool you can use when you have a multi-omic RNA plus a TAC-seq dataset is to correlate peaks with gene expression. We're going to create a new analysis using all genes, look in a particular range around the start of every gene, and then run this tool. When the results come back, you'll see a table of genes and peaks that are correlated along with the z-scores and p-values. You can filter for specific genes like so, or you can jump to the attack browser to view any of these features. So let's focus on this one, SLC12A2, and this peak on chromosome 18 that appears to show a correlation. We'll click to view in the attack browser. You can see that this peak is enriched in glomerular endothelial cells. We'll start zooming out. I don't see that gene yet, but as we get farther out, we will eventually see that on the right-hand side over here. It's all the way over here. So this shows that you can find peaks that appear to be correlated with RNA expression, even from a distance. Thanks for watching our guide on what's new in version 2 of Cellismo. We'd love for you to try it out and let us know what you think.